talk very briefly about this matter of anchors. Simply really just to list some of the problems that seem to affect the study of biologic effects and exposure to electromagnetic fields. These include the problems of accurate individual measurement as opposed to field measurements, the problem of exposure gradient. If you don't find a big gradient of exposure from those with small exposure to large exposure, you're going to have a lot of trouble. Of course, if everybody gets exactly the same exposure, there's no way in which you can demonstrate from human studies that that exposure is causing some bad effect. Sample sizes tend to be small. I've run across this quite recently with some work on the power line now under construction in New England. There just aren't going to be enough people living close to that line to do the kinds of studies that one might want to do over a period of years to see if their health is affected. There are the problems of poorly specified hypotheses for study. They're not really nailed down ahead of time the way we'd like to have. Individual heterogeneity is a problem. Not everybody responds to these things the same way, and you may need to consider that in trying to design studies and then interpret them. Confounders, it's a technical term in epidemiology. You might think of them as risk factors other than the exposure under study, and other sources of bias in the data can be a problem. And finally, the problem of what statisticians call multiple comparisons, including types of fields, ranges of frequencies, and energy levels. If you go out and look at 100 different health endpoints and examine each one at the usual statistical level of p less than 0.05, you expect to find 5 significant results just by chance alone. And it would be very hard to, if you find 5, it would be very hard to conclude from that alone that there's really anything going on. I might point out that this applies to confirmatory studies as well. If you've got 1 chance in 20 of getting a statistically significant result the first time by chance alone, then 1 time out of 20, a confirmatory study is going to tell you the same thing. And so that even a single confirmatory study may often not be quite enough. Some possible consideration. My third point was the need to have a way to set priorities. I've just started to list some of the things that may be important in this. How strong is the present evidence that there may be an effect? How large is that effect likely to be? How many people are affected? All of these things should affect priorities for new work. How good is the opening for new advances? If nobody really has any good proposals, that ought to be reflected. What will new information contribute to the overall evidence? Are you filling in the 4th decimal point? Or are you really breaking new ground? Who is affected, including synergies? I've talked about that before. Are the victims of a particular exposure identifiable in some way? Can you tell who individually was affected and who wasn't? It's much harder to get ahead if they're not identifiable. Let me illustrate. Victims of accidents are generally identifiable. You know who was injured. Victims of exposure to a carcinogen are generally not identifiable. You know that some class of persons as a whole has an increased risk. You may know that some worker population, for example, has 3 times as many lung cancers as they ought to have, but you expect some anyway, and you can't point to individuals and say that one but not that one. Finally, I'm not suggesting that everything be locked into priorities. Do not commit the whole of the available resources to any set of rigid priorities. Preserve the flexibility that investigators need, the most innovative investigators. I'm just finishing. Probably put the majority of your resources into some kind of priority system. That last point, of course, is more advice to the funding agencies than to the investigators themselves. Thank you very much.
Our next speaker, as you already know, is Dr. William Ross 80, a native of Adelaide, Australia. Received Bachelor of Medicine, Bachelor of Surgery, and MD degrees from the University of Adelaide. Dr. 80 is presently associated with the PES Memorial Hospital at Loma Linda, where he is Associate Chief of Staff for Research and Development. Since 1977, he has been Professor of Physiology and Surgery at Loma Linda University School of Medicine. Dr. 80 has a long list of credits and honors that you can read about in the program. And not wishing to take any more time from the important presentations, I give you Dr. William 80.
Thank you, Mr. Chairman. I'd first like to express my deep gratitude at the honor of being a member of this panel this morning, recognizing that it is one of those rare foci where people from many different disciplines converge on this rather troubling question of health-related effects of environmental electromagnetic fields. May I interpose here at this point that if it's agreeable to those concerned, I would suggest we meet this afternoon at 1:30 rather than 2 o'clock, since I have a 4 o'clock plane. That's in the 8th floor conference room of the Health Services building opposite. What I would like to do there is to show two videos, one of which is 15 minutes long, made at Disney Studios on cell membranes and cellular communication. The second is a very interesting BBC video, which I was privileged to help orchestrate. It's a panorama program. That is their equivalent of our CBS 60 Minutes. They deal with one topic every Sunday night. This one is called Electricity, a Shock in Store ? And it lasts for 40 minutes. I think you'll find it of extreme interest, and would be very pleased if you can find it possible to be present. Now, this morning, my task is to present a perspective on cell and molecular mechanisms that couple weak environmental electromagnetic fields to tissues, and the evidence that these interactions relate to cancer promotion. In 25 minutes, I can hope only to offer pointers to the major research accomplishments and creative achievements of highly qualified researchers in this country and abroad, from whose work we now have a reasonably connected picture of the sequence and the energetics in the major steps that couple weak environmental electromagnetic fields to living tissues. And this work should not be the plaything of vested interests. In such a short presentation, there is a grave risk of trivializing the importance of a great revolution in biological concepts that has taken place solely through the use of imposed electromagnetic fields as tools to study the atomic rather than the molecular aspects of living matter. A revolution probably greater than any other in the history of biology over the last 600 years. A revolution that now points the way in cellular mechanisms to a completely new understanding of the essential steps in tumor formation. Could I have the first slide, please? Yeah, I think we can dip those top lights. Thank you. Now, since we don't have a pointer, I'm going to ask you to look at this diagram, which shows a rather odd-shaped device, which is a nerve cell. It has within it a blob that you can see as a nucleus. Dr. Sagan referred to that as the site of damage to its store of DNA, which I think everybody knows is the body's mechanism for storing genetic information. But it is enclosed by a membrane, which I've drawn here as a thin line, and you can see other cells around this particular one with dotted interiors. They are a different sort of cell. This is part of brain tissue. But what I draw your attention to is that one cell, enclosed by a membrane, is separated from others by little gutters. And these narrow gutters, so-called intercellular spaces, are the preferred pathways for the passage of current through tissue. The reason being that cell membranes have very high resistances. We can measure them at, say, up to 100,000 ohms per square centimeter, whereas the fluid between cells has a specific resistance of the order of 50 ohm centimeters, or roughly 1,000 to 10,000 times better as a pathway for current in tissue, and intrinsic currents generated by tissue or fields from outside the body travel mainly through these gutters. Also, as we shall see with respect to the cancer model, the question of communication between cells is now seriously raised with good experimental evidence that says that if we interrupt the communication between cells, the result is uncontrolled, unregulated growth. In other words, this whispering together by signals that are both weak electrical and chemical is an intrinsic aspect of tissue organization. The cell membrane is thus a device which is a sensor and an electrical barrier, and is a means of communicating signals from outside to inside the cell. I regret that we do not have a pointer, but I think I can explain what's going on here. The very thin layer of the membrane, seen here as the sweeping swath that crosses the slide from below here on the left to the top right, is a double layer of fat molecules 1/millionth of an inch thick. That 1/millionth of an inch supports an electric gradient of 1/10 of a volt, or translated into more familiar terms, 100,000 volts per inch. That is the nature of the electrical barrier of every cell in our body. And if you think of it from the engineering point of view, it would be very difficult indeed to have an insulator that would stand that level of dielectric gradient without breakdown. But that is, in fact, the nature of the process, and that is the status of the cell from birth to death. Now, interposed in that double layer of fat molecules, you can see some little projections or strands that cross it. Those are particles of protein, strands of protein, which I will show later, and on the outside of the double fat layer, they spread out into a forest of branches, highly electrically charged negatively, and thus attracting to the surface of the cell calcium and hydrogen ions. So we see here a membrane which has a lot of Trojan horses in it. Those Trojan horses, with their external branching arborizations, as we call them, form receptors for hormones, antibodies, and nerve transmitter molecules. They act as pathways from the outside of the membrane to the interior. And then on the inside of the membrane, you can see a number of large objects which are enzymes. Enzymes are protein catalysts and can carry out functions as any other catalyst does, just like the catalytic converter in an automobile, entering into chemical reactions, carrying them through in an enhanced way, and emerging from the reaction unchanged, so they can go through the same series of reactions over and over again. And the burden of the work that has gone on with electromagnetic fields has been to show that the enzymes can respond to very weak electric fields on the outside of the membrane. Now, when we talk about the sensitivities of these systems, here are some of the observed sensitivities against which we can set some of the work concerned with electric power lines and environmental fields. It is now accepted knowledge, unquestioned knowledge in the biological community, that things like the navigation of sharks and rays, the daily rhythms in birds and man, the estimation of time in monkeys and man, and the control of circadian rhythms, is influenced by fields in the tissue down to the level of a 10/millionth of a volt per centimeter. And at the same time, most of those sensitivities relate to fields below 100 hertz. And if you look in the lower part of the slide, you'll see there a set of biological constants. I mentioned that the membrane potential is a barrier of 100,000 volts per centimeter, or roughly 100,000 volts per inch. The two lower things, the synaptic potential and the electroencephalogram, give a measure of change in the membrane potential. The electroencephalogram, the EEG, which we're all familiar with, recorded on the surface of the head, if we measure it at the surface of the cell, that is a gradient of 1/10 of a volt per centimeter, or 1/1,000,000th of the membrane potential. And it is representative of most of the electric oscillations that occur in tissue, which until recently had been said to be only the noise of the process. The EEG was described as the noise of the brain's motor. So it was thought that these things couldn't do that these intrinsic fields, or fields from outside that were at the same level, could not have biological significance. And the naysayers came from three directions. The engineer said, if it can't fry you, it can't hurt you. The physicist said, the energy of these fields is not strong enough to break a hydrogen bond, and therefore in concepts of thermodynamic equilibria, there can be no significance to them. The physiologist said that the field energies are too low to alter the ionic equilibria. That is, the fact that in the normal state of a resting cell there is a lot of potassium inside and a lot of sodium outside, and this field is too weak to alter that energy, that equilibrium. These three points of view are erroneous, and they are erroneous because they overlook cooperative organization, in which there are coherent states between molecules and portions of molecules where charges see one another resonantly. And this is not a matter of speculation, and it is not a matter of half knowledge, because you and I would not be here talking if we did not have in the red cells in our body the hemoglobin, which sees at an iron atom something resonantly on a protein molecule adjacent to it 30 angstroms away, a galactic distance. And those two interactions occurring resonantly allow the hemoglobin molecule to pick up oxygen in the lungs at a slightly alkaline pH and unload it in the tissue at a slightly acid pH. And if we take the iron heme away from the globin, we lose that cooperative interaction. The eye can see down to 1 photon of energy at the blue-green wavelength. The ear can hear down to the level of the Brownian movement of the molecules of the inner ear fluid, and every one of these processes depends upon an extremely high degree of cooperativity. Now, I wanted to deal briefly with the essential steps in transductive coupling, as it's called, the sequence of events that occur between the outside and the inside of the cell. Oh, we can skip over this. This was discussed earlier. The sequence from the outside to the inside of the cell is firstly an alteration in the binding of calcium to those arborizations, the tree branches. And that is very, very cooperative. A very weak event can produce a very large alteration in calcium binding. And the second event is the coupling of that signal down the protein strand to the cell interior, and the third event will be the activation of those enzymes on the inside of the membrane. Let's look at those things in a swift sequence. First of all, if we look at the binding of calcium to the cell surface, when it is manipulated by applied fields, either a radio frequency field that has low frequency modulation on it, that is the top set of curves, or strictly low-frequency electric fields in the bottom set of the bottom graph, we see that the calcium is modified in its binding. It is decreased in the upper curve where the signal is 1/10 of a volt per centimeter. It is increased in binding where the signal is around 10 to the minus 7 volts per centimeter. But the two curves rather strictly mirror one another. And this is the measure of the amount of calcium that comes out of brain tissue when we expose it either to the weak radio signal above or the far weaker electric signal below. I draw your attention to the fact that this is a highly windowed phenomenon. In other words, it occurs only at certain frequencies. And the measure of this cooperativity is the fact that there are windowed interactions. Windows with respect to frequency, windows with respect to time, and windows with respect to intensity. And the calcium binding is also sensitive to the intensity of the field. More is not better in this respect. You get a threshold, but increasing the intensity causes the response to decrease. So we see here windows in frequency, windows in intensity. I will show you a number of other windows with respect to highly complex cell processes in the course of the next few minutes. And it is not true, as was stated in the EPRI journal for October-November last year, that there are a few studies which have suggested that effects are more pronounced in certain frequency windows or bands. There are not a few, there are many. These windowed phenomena are at the very essence of the way in which weak electromagnetic fields interact with biomolecular systems, and we can measure them not by the few, but by the dozens. The transmembrane signal that runs down from those, let me exemplify that last point. First of all, if you take embryonic bone, as was done by Fitzsimmons in a paper published in Biochemica Biophysica Acta, and you expose it to an electric field, not a magnetic field, capacitively coupled, a field in the range of 10 to the minus 7, 10 to the minus 8 volts per centimeter at 10 hertz, you find that there is the release of a hormone, an autohormone, necessary for bone formation, and that is a comparison of the effects of, or rather the release of that hormone in a control culture of bone, embryonic bone, and the effects of the weak field. And you can see that even down at 10 to the minus 7 volts per centimeter, there is a highly statistically significant modification of the release of that hormone. It's called skeletal growth factor, and it is a calcium-dependent release of that hormone. So I want to exemplify the fact that windows abound in this type of interaction. Now, the transmembrane signal runs along a segment of protein into the interior of the cell, and the fascinating aspect of that signal is that it is mediated by calcium, but the protein that carries it is non-conducting. And this was the basis of, or part of the reason for the award of the Nobel Prize in Medicine in 1987 to Cohen and Levi Montalcini, because they investigated the nature of the transmembrane signal down these protein strands. I don't have time to go into it further this morning. But since it is not electrically conducting, there have been many thoughts about the way in which it could occur. And one of the strong suggestions is that it is mediated by vibration modes in the backbones of the protein. You see in this, my drawing here, that there is a triple stranding of the backbone. And the vibration mode produces what is called a soliton wave. The hunt is on for soliton waves in these proteins. And the effect is one that was first modeled over 100 years ago in water waves, where soliton waves can pass for very long distances uninterrupted by surrounding oscillations in water. Here you see a Nautilus-class submarine on the surface generating a whole series of solitons as it passes. And in fact, though I'm not privy to any aspect of the classified information, there is the possibility of determining the position of submarines at great distances by the use of, or by the detection of soliton waves. And the work in the biology of these soliton systems is ironically by people at Los Alamos, Alwyn Scott's group, and Davidoff presumably engaged in the same type of work in the Soviet Union, leading to the biological model of this process known as the Davidoff-Scott soliton. The question of soliton formation, for those of you who may be more familiar with the physics, involves the addition of energy to a system which has a large number of oscillations which may not be bound together in a highly rhythmic fashion called pseudo-periodicities, and one goes through processes of entrainment and the bifurcations leading to chaos, and finally the destabilization of the chaotic quasi-periodicity and the formation of a soliton. Now, the question of coupling to intracellular enzymes is leading us in the direction of the cancer promotion process. The studies of weak field interactions with enzymes has led to findings that there are at least 3 groups of enzymes sensitive to electric and electromagnetic fields on the cell surface. The first is metabolic, that large lump that you see there inside the cell membrane, but attached to it is adenylate cyclase that takes the fuel, adenosine triphosphate, converts it into another substance, a cyclic adenosine monophosphate, releases large amounts of heat energy in metabolic processes. That enzyme is sensitive to many different types of weak electromagnetic fields. To the right of that large lump, you can see some little blue dots or circles. They are protein kinases, which are activated. They are messenger enzymes. They are activated by the formation of the cyclic AMP from the fuel substance adenosine triphosphate. And then up on the top right, you see yet another enzyme, which is ornithine decarboxylase, ODC for short, essential for growth in all cells. It is active in cancer cells, and all cancer-promoting substances turn it on, but not everything that turns on ornithine decarboxylase is a cancer promoter. However, the interest in it is that it is necessary for the synthesis of DNA and certain proteins. So we have 3 groups of enzymes sensitive to what is going on outside the cell. They are metabolic, messenger, and growth enzymes. Now, the question of the let me be sure I keep the trend here. The question of the messenger function has been investigated in the lymphocytes of the blood. And here you see a graph of the sensitivity of lymphocytes, part of the immune system, to a weak microwave signal that's sine wave modulated at the frequencies that are along the abscissa there from 0 to 100 hertz. And you see that the lymphocytes only respond in a windowed fashion, and the maximum sensitivity is with 60 hertz modulation, 60 hertz modulation. Nothing below that, and gradually decreasing at higher frequencies. The same system of essential enzymes in the lymphocytes is sensitive to the time of exposure. Here is a window in time. They are exposed continuously for 1 hour, but they respond only for in the first 15 to 30 minutes of exposure. Now, this raises a very significant point in the potential relationship of these laboratory findings to the epidemiological problem. What if the immunological system is affected only by intermittent exposure, when you first enter the field or when you come away from it? And that leads to the question of cumulative dose. Those who come to this work from ionizing radiation tend to presume, I submit rather arrogantly, that they know all about cumulative dose. They don't. The point is that for ionizing radiation, you can keep on adding and adding and adding, and you do get the total effect of the ionizing radiation. With non-ionizing fields, you do not. And there are so many on and off effects that one must question the significance of total exposure time, whether it be in the home or in industry. Now, the question of I now want to bring in the next topic, which is, what is the evidence relating cell membrane processes to cancer promotion? What is the model of tumor formation to which weak electromagnetic field action relates? You've heard a great deal this morning about epidemiology, and the use of animal models is rather difficult, very expensive, and certainly far below that which is needed for an adequate measure of whether or not fields can alter growth and can play a role in cancer promotion. However, in Sweden in the last year, 2 studies have been published with respect to VDT fields, the computer terminals, using animal models, and this is a slide from one of them where mice were used, exposed to a 20 kilohertz VDT type field, and you see here at the top a mouse with a brain that has no skull, and then on the bottom left the mouse has protruding abdominal viscera. There is also a spina bifida, which means that the spine is open behind, and a number of other major abnormalities. I forget the total details. The one on the right has also spinal defects and a modification in the rib formations. These studies showed increased fetal death, increased fetal resorption, and one study showed an increased incidence of these abnormalities, the actual teratological abnormality. The question of the tumor formation itself and the question of fields and tumor formation leads us to look at 2 different aspects of the interaction. One involves the immune system, the other involves a direct effect on growth. Now, the immune system is the guardian against abnormal cell development and the recognition of cells that do not belong in the body. And the presence of tumor cells leads to a very powerful killing by lymphocytes of the cells of the immune system in what is called allogeneic cytotoxicity. What it really means is that the lymphocytes go on a hunt in a Pac-Man fashion. And what this graph shows is the capability of lymphocytes to kill tumor cells. And you see here the reduction in killing capacity when lymphocytes were pre-treated with a weak microwave field modulated at 60 hertz. And the killing capacity was reduced by about 25%. It is windowed. You see that at all the other frequencies tested other than 60 hertz, the effect was less. And this study was replicated with 60 hertz fields at 10 millivolts per centimeter, 1 millivolt per centimeter, and 0.1. At the time this graph was made, the 0.1 level was not complete, but let me tell you what has happened there. At 10 millivolts per centimeter, there was a 30% inhibition of killing. At 1 millivolt per centimeter, 18%, and at 0.1 millivolt per centimeter, it was 7%. The 1 and 10 millivolt levels were highly significant. The 0.1 millivolt was statistically insignificant. It was merely a trend. Now, in terms of field levels in tissues from high voltage fields, around 0.1 millivolt per centimeter is a level not atypical of being close to a high voltage power line. So here is the loss of killing capacity by these lymphocytes. The question of tumor models per se has only been indirectly addressed here this morning, and I think we need to talk to it more directly for a moment. Carcinogenesis is now viewed as a 2-step process. There is initiation, which is the damage to the DNA that Dr. Sagan spoke of, but that is only the first step in the process. The second is promotion, in which cells with damaged DNA form a tumor. So we speak of initiation and promotion. The initiation involving DNA damage produces a mutated cell which transmits this mutation to daughter cells, but a cell with damaged DNA, an initiated cell, may never produce a tumor. May never produce a tumor. We all have perhaps millions of initiated cells. The promotion process is separate and later, and it involves the action of substances which are chemical promoters, do not produce mutation, and electromagnetic fields likewise, as Dr. Sagan said, do not produce mutation, but they are, from many studies, now showing effects of being promoters and acting jointly with chemical substances that are known to be promoters. Now, this slide shows a whole series of promoters. Some, like the tobacco extracts, are both initiators and promoters, but it's been possible to isolate from tobacco proteins that are not initiators but are very powerful promoters. Both the fields and the promoting substances appear to act at cell membranes. Now, given that there is this evidence, which I will present very briefly in a moment, for joint action of environmental chemical pollutants with electromagnetic fields, one may ask, what is the value of doing epidemiological studies that do not consider the joint exposure to chemical pollutants? And if you're going to go into the chemical environment, you immediately enter a world of worms where you may never know what it is that was involved. And I would suggest that the value of epidemiological studies is rather limited in the context of the very complicated lives that we lead, both electromagnetically and chemically. The question of the dose-response relationships that Dr. Bela mentioned also come into this question because, as I mentioned a moment ago, you cannot estimate the effect of an electromagnetic field, a non-ionizing field, simply by saying you were in it for an hour. There are other factors that relate to the on effect and the on effect. So let's look briefly at cancer promotions mechanisms in the cells and the fields. The two things that I want to address here are the actions of chemical cancer promoters and fields together on cells, on the enzymes, and then I want to speak briefly to the disruption of intercellular communication. Now, here is the effects of 60-cycle fields ranging in intensity from 10 millivolts per centimeter, which is high, down to a tenth of a millivolt per centimeter. And what one finds here is a windowed effect, a very strong effect at 10 millivolts, a very strong effect at a tenth of a millivolt, but in between those two levels, very little effect on the enzyme activity. This is ornithine decarboxylase, the enzyme essential for cell growth, and the enzyme which is now used clinically as a measure of whether you may have cancer of the prostate or not. Now, in an audience that is predominantly male, this should be of abiding concern, because if you look at the incidence of cancer of the prostate, every one of us will have it by the age of 92. And given that alone as a consideration, it is important to know how we may estimate its presence, and medically it is more reliable to measure the activity of this enzyme than to look at the cytology, the appearance of the cells. Now, that was with 60 cycle fields. Here we see the joint actions, or the separate and joint actions, firstly in the red graph, of a powerful cancer promoter. It's called a 4-bol ester. It comes from the euphorbia plants. In China, it causes cancer of the nasopharynx in people who eat it. And the green graph, shows the enhanced action on the ODC enzyme of the weak microwave field modulated at 16 hertz together with this 4-bol ester. So you see there is a very powerful enhancement. Parenthetically, I would point out that this work by my colleague Dr. Craig Bias has been published in 2 of the leading cancer journals of the world, Cancer Research and Carcinogenesis, a point to which I shall return in a moment. Now, the question of what What we now know about the disruption of intercellular communication leads us to the awareness that has come to the cancer field in the last 2 years, not to this area of bioelectromagnetics, that cells communicate with one another through gap junctions. And that gap junction system is a little plaque of protein that has tubes through it. It mediates electrical communication, it mediates chemical communication. Cells make substances needed by their neighbors, and to disrupt that leads to a loss of the health of a community of cells. And the work from the cancer field shows that disruption of communication through that little plaque of protein leads to unregulated growth in many types of cell cultures. Also, if one takes a culture of cancer cells and puts into that culture normal cells, wherever the normal cells make contact with the cancer cells, growth ceases, and in many cases, the cancer cells differentiate again into normal cells. Now, to those of us who went through medical school many, many years ago, that is either a heresy or a miracle, depending on your point of view. But the fact that cancer cells can be turned back into normal cells has become an abiding consideration in the way we look at the problem of cancer, as I shall explain in my last remarks. The question of how cells communicate, whether they are normal cells or cancer cells, is exemplified in the following slides. This is a culture of normal ovarian cells, and you cannot see it in this phase contrast diagram, but one of them, and I can't point it out, has been injected inside with a dye. And this is the same culture, the same scene, now seen after digital processing of the image to show what has happened to the dye. The cell that was injected is to the right of center and has two little white blobs in it. But notice that it is surrounded by many other cells that have received the dye through gap junction mechanisms. Now let's look at some cancer cells by contrast. Here are malignant melanoma cells. And 3 of those cells have been separately injected inside themselves with dye. And here is the scene in the digitally processed image. The dye has remained within the cells and has not transferred itself to any other cell. Now, the question of whether or not one can open gap junctions with cancer-promoting substances has been a focus of much research in the cancer field. And this diagram shows how the opening of the gap junctions allows the entry of killer substances into cells. LT is a lymphotoxin which will kill cells by disrupting their membranes. That process of opening gap junctions in the presence of the cancer-promoting substances is enhanced by electromagnetic fields. Here's a slide that shows the effect of TPA, that is the cancer-promoting chemical, with and without electromagnetic fields imposed. The field was a weak 16 Hertz modulated microwave field, and you can see in the third bar, the open bar, that there is a very marked effect of the microwave field in enhancing the action of the cancer promoter. Now, the work that I have described here is something that has come as much from cancer research as it has from our field of bioelectromagnetics. And it has led to the development of a new area, a new field in cancer research, which has this rather awesome title of non-genotoxic carcinogenesis, meaning that it does not involve the DNA or the genes in the first instance. It is work that has occurred in laboratories worldwide and under the best circumstances and done by the best people. So we do not come with half-baked notions to this area of the role of cell membranes in cancer promotion. The key paper in that book is one by Yamasaki in Paris, The Role of Cell-to-Cell Communication in Tumor Promotion, and his opening remark is that cancer is a disease of the society of cells, and that the disruption of cell-to-cell communication is the key element in tumor formation. Now, I'm going to end my remarks there and emphasize that in order to understand these processes, we must tackle the problem from the point of view of joint actions of chemical substances and electromagnetic fields, and we must examine the problem from a physical rather than a chemical point of view. In less than a century, the frontiers of biology have moved from organs to tissues to cells to molecules that are the exquisite fabric of living matter. Now we stand at a new frontier. It may be more difficult to understand, but is of vastly greater significance. It is at the atomic level, rather than the molecular, that physical rather than chemical processes now appear to shape the flow of signals and the patterns of organization in living tissue. Thank you.